All right, thank you for being here. I'm Naila. I'm a senior PhD student, and I study a mouse model of Parkinson's. But today I'm just going to talk a little about a project I've been working on with a few other people, quite a group, to look at how we can translate some research, for instance, for Parkinson's disease into music and dance. And I want to thank Anusha right away. I don't know if you want to get up or like raise your hand or something. For one, composing all the music that I'm going to show, and two, also always having a good pen up her sleeve for titles. So I study Parkinson's disease. It's a neurodegenerative disorder. It's kind of diagnosed and characterized by these changes in motor function, particularly like stooped posture, rigidity, slowness of movement, facial masking. And then there's a number of non-motor symptoms that also really impact quality of life. And it's important to note that there's currently only really treatments that mitigate the motor factors, the motor symptoms, but don't really address any of the non-motor symptoms, and also not disease progression. The cause of Parkinson's is kind of unknown, and it can differ from one person to the next, but it's usually a mix of both environmental factors and genetic factors. And there are a number of different gene mutations linked to Parkinson's. In the lab, I study the LRRK2 gene mutation, which alters the LRRK2 protein, but I'll also be mentioning the SNCA mutation, so that's a different gene altering a different protein, and the VPS35 mutation and its effects on the VPS35 protein. So all of these genes and their mutations change the function of proteins in the brain and in the body, and they do a lot of crazy things in complex diagrams, which I'm not going to talk about. Instead, I'd like to present it in the form of music. And I have not trained rats, unfortunately, to play instruments. If you have ideas on how to do this, I'm open. But instead, we're just going to look at, very simply, how do we represent gene mutations in music. So this is not really my novel idea. A lot of people have been experimenting with how to look at DNA sequences in music. And I even found a study that used music to represent different protein sequences and how you could hear the protein mutations better than you can actually detect them, say, in a sequence. So it's kind of playing on this idea that we're much more in tune to listening to differences than we are in seeing them in a code. So that was kind of also a basis for this project. So for our project, we started, or I should say Anusha started, by just representing a DNA sequence in a melody, which I'll briefly play. So this is just a very simple, a very nice, simple piano piece that just represents a basic template of a DNA sequence. And on top of that, we looked at how codons, which are triplets of nucleotides, each code for a different amino acid. So there's 20 amino acids, they're the building blocks of proteins, and you can take like 3 bits of DNA, which code for 1 amino acid. So we kind of created a harmony that goes over top of the melody that represents in triplets the amino acid sequences. So when we put it together, does it stop? Oh, yes, it does stop. So we have the genome, which is the melody, and the proteome, which is the amino acid sequence, and we have a melody and the harmony over top. So this will become very familiar sound to you. I'm going to leave it playing so that it really embeds in your mind, so that when we add the gene mutations, you can hear the difference. So we'll just have this playing in the background while I explain the PD mutations, Parkinson's mutations. The first one is the LRRK2 mutation, so this is what I'm studying. It's a point mutation, which means that you're switching one nucleotide for another. So the DNA mutation is very specific. Within the LRRK2 gene, it's just switching a nucleotide for another nucleotide, which in terms of music would be switching one note for another. And the effect this has on the amino acids is that you're changing one of the nucleotides in the triplet. So instead of coding for one amino acid, it's going to code for a different amino acid. So that kind of messes up the resulting protein. In that spot, there's a change in the protein formation, and so the protein function is altered. VPS35 is also a point mutation, so same thing, you're substituting one nucleotide for another, so in this case one music note for another, And again, the amino acid sequence is altered. So in the triplets that are playing in the harmony, we also alter a note. And then the last one is SNCA. So this codes for a protein called alpha-synuclein, which is also altered in Parkinson's. And this is either a duplication or triplication. So the gene sequence, or the little DNA sequence that codes for that protein, is repeated several times in the DNA sequence. And so that results in more amino acid being produced and more protein being produced. So kind of causes like more aggregates in the brain of alpha-synuclein because there's more protein and it's overactive. So I'm gonna play just the melody because it's the clearest to hear the changes in the melody. So the first loop will just be the basic melody that we already had, and then each loop is adding in one of these mutations. Oh, no, that's not what I meant to do. So this is just the bare melody, the one that you've been hearing for a while now. This is the awkward part, right? Try that. So now it's going to loop a second time, and there's one note that will be different for Lerc 2. It's coming up. Oh. Yeah, we're going to speed this up. Okay, maybe I'm going to play the... So now you'll hear the Lerc 2 mutation, so that same note altered. But there's also the D, which I just talked over, which was the VPS 35 mutation. You'll hear it again. And that was the Lerc 2. And then the third time around starts now. So that's the triplication. And I'm going to stop this and play just the whole piece with the harmony above it, so you can hear it. You can hear, you can't really hear it distinctly, but the amino acids have also been switched, so in the corresponding spot where the mutation is. And that's the first part of the project. Just to finish up, so the next step is to expand on this music for the second part, which will be looking at physiological changes in the brain. So we're coding like different protein interactions or how neurotransmitters are altered or how the disease progresses into music and kind of building on the first melody we had to express more neuroscientific research. And then the third level is the human experience. So looking at the diagnostic symptoms, or in this case, the motor symptoms, and allowing that to change, for instance, the rhythm of the piece. So kind of trying to mirror the motor symptoms and how it alters movement into the music. And then we're also creating a dance for this. So it might be hard to hear just the mutations, but if you have a visual representation, it's a lot clearer. So we want to have a dance for the genetic level, the physiological level, and the human level. And we want to do the same thing for other movement disorders. So the whole project is looking at different movement disorders, their genetic basis, the physiological effect, and then obviously the lived experience, bringing in people who have these movement disorders to kind of inform the piece and perform with us. And this is a part of a team project, the Peace of Mind team, which is kind of focused on performance and science collaborations. And if you're interested in joining us for this piece or another piece, please come talk to me. I'm done talking now. Too much.